So in the 1800s, Darwin first wrote about this idea of evolution caused by natural selection or survival and reproduction of the fittest, saying that it led to changes over time in organisms, and in his famous work on the origin of the species. But we need to be really clear about what is doing that evolving. So my question here is, does an individual evolve during its lifetime? And so hopefully you can answer that as no. So just because I would survive better if I had a longer finger, for example, let's say I'm an animal that gets food by using fingers, even if that would lead to my improved survival, I can't choose to have a longer finger. I can't cause my finger to be longer and therefore survive longer. Now, what happens with survival of the fittest is that if I have some offspring that have variation in their finger length, then of my offspring, the ones with the longer fingers would be more likely to survive and reproduce. And therefore, over many generations, we would see selection for the long-fingered trait. So when we are talking about evolution, we're not talking about individuals evolving. We're talking about groups of organisms. And this is because we're really talking about many generations. So groups of organisms evolve. And there's a formal term for that, the groups. Populations are groups, the groups that we're talking about. So while individuals are selected for through natural selection, so we're not saying that there aren't effects on individuals, it's actually populations that evolve. And again, that's groups of organisms. So those are groups of individuals that are of the same species, they occupy the same environment, and they can interbreed with each other. So depending on the organism, the population can be a very small geographic area. So imagine this is my continent. For some organisms, I might have population 1, population 2, population 3, population 4. For other organisms, this whole area might be one population. So for example, this might be my population of deer, but for rabbits, I have 4 different populations. So why would this same geographic area have only one population of deer but 4 populations of rabbits? It really depends on the ability for them to reproduce with each other. So deer migrate, and because they migrate and move long distances, they're able to have deer from here end up breeding with deer over here. And so that keeps them the same population. Rabbits do not migrate as a routine part of their life cycle. They can't travel as far, so you might expect that you have a distinct rabbit population over in this valley and over in this desert, and they don't travel between these 2 areas, and so they're not breeding with each other. And so even though they're the same species, they are different populations. Okay, so population is not equivalent to, it's not the same thing as a species. The same species can actually be divided into multiple populations depending on the type of organism. So now that we've just defined the term population, let's talk about population genetics. So population genetics is looking at the different genes and the different genotypes that are present in a particular population. So this helps us understand how much genetic variation there is, and then what's causing it and why it's staying there or not staying there, depending on the population and what's happening to it. So it's tracking changes over time in the genetic makeup of a group. Just another more simple way to put it. And then the genetic variation is related to phenotypic variation. So this will also tell us about physical trait variation as well. So for many, many traits, the genes that control them are polymorphic. Polymorphic means many forms. So what we're saying is that for a lot of traits, there's variation. So if we look at these flowers here, so see across this field, we're seeing different colors of flowers. So that tells us that flower color is polymorphic in these plants. Okay, so a polymorphic gene can have 2 or more alleles. You hopefully remember this from Mendelian genetics. So for example, we used big A allele or little a allele. So different variants of a gene. Okay. So we've said that many genes are polymorphic. That means they take many forms. Some genes are monomorphic. That means that there's only a single allele, or most individuals have a single allele. We also use the term fixed. That means about 100% of individuals have that allele. So if you think about like really essential traits, like for example, having 2 arms, right? Humans have 2 arms. And in fact, this is a trait that goes back very, very, very far in our to our ancient relatives having 2 arms. And so that trait is really fixed. Almost 100% of individuals genetically are wired to have 2 arms, not 3 arms, not 1 arm, not 6 arms, but 2 arms. So we're not saying that every trait has variation, but that some do have 100% fixation of a trait. But for many, many traits, again, there's polymorphisms, there's allele diversity, and genetic diversity is going to be what we're measuring when we look at population genetics. And genetic diversity is important when it's possible, right? So for these polymorphic traits, genetic diversity is generally a good thing because it's the raw material of evolution. So remember, you need variation for evolution to occur. That's what we mean when we say that it's the raw material. If everybody has exactly the same genes, then there's no benefit to any particular trait. And it might be a bad trait, but nobody has a good one, right? So you're not going to get evolution. So the gene pool of a population, so if I'm doing population genetics, I want to understand the genes and alleles that are present in a population. That's measuring its gene pool. So the gene pool is all of the alleles for every gene in a given population at any one time, right? So you're comparing it like now versus in the past, or now versus in the future. And so we measure the gene pool using allele frequencies and genotype frequencies. So 2 different measurements. And again, these are going to be values. They're going to be numeric values because they're measurements. So in order to understand what these terms mean, let's just refresh again about what an allele, a genotype, and a frequency is. So alleles are variant forms of a gene. So again, kind of tapping into your memory of Mendelian genetics. Remember we had, for example, an A allele that was dominant and an A allele that was recessive. Or for flower height, there was tall versus dwarf. There was purple flowers versus white flowers. So we had like a dominant allele and a recessive allele in the case of a lot of the traits we looked at for Mendelian genetics. The genotype is the combination of alleles that an individual has. So for example, you might have individuals that have a dominant and a recessive, or 2 dominants, or 2 recessive. I'm just going to move over here because I also want to remind you of some other terms. So remember homozygous and heterozygous. And then we could have homozygous dominant, or we could have homozygous recessive. So these are verbal ways to describe genotypes. So homozygous means 2 of the same, which is why it's either homozygous dominant or homozygous recessive. So 2 of the same dominant, 2 of the same recessive. Heterozygous means 2 different. So this would be a heterozygous genotype. So those are verbal ways to describe genotypes. And then the last term here, frequency. A frequency can be compared to a percentage. It's telling you how much out of a total. So we might have, for example, 5 of one particular thing out of 10 total possible options. So think back on maybe like tossing or flipping coins, for example. So you might have 5 heads out of 10 possible times you flipped the coin. Or in the case of genotype and allele frequencies, you might have 5 of the little a's out of 10 total alleles. And this is why you could compare it to percentage, right? 100% is the total, and you say, okay, I have 20% of, 20% red shirts out of 100 total shirts. But when we use frequencies, it's a different value. Instead of a percentage, it comes out of 1 total. So the bottom will always add up to 1 for frequency instead of 100%. So touching on math here, basically, the math difference between a frequency versus a percentage is that the total is 1 instead of 100%. And so you'll have decimals. So this will lead us to have decimal values instead of whole number values. Again, hopefully touching into your memory of frequency versus percentage from math.